But just to review, last time in class we were talking about the Clean Water Act and this idea of biotic integrity, kind of how we measure that. And as you all correctly put on today's celebration, this Clean Water Act, there, I'm gonna untangle that later, was really good at eliminating a lot of point source pollution, particularly through these grant programs to municipal wastewater treatment facilities. But then we realized our water still was not all that awesome. And so then there was this focus on, look, let's look at the actual, what we might call the emergent properties of the streams. So what is there as a result of the water? So instead of like measuring water chemistry constantly, instead, let's just look at what is there because of the chemistry. And then this focus on wet weather pollution, this non-point pollution, which is such a more intractable problem. It's a lot harder to solve than, fixing up your wastewater treatment plants. And of course, this then is where we relate to macroinvertebrates, is with this idea of biological integrity. And as we talked the other day, you can look at this with fish, macroinvertebrates, algae. There's probably other stuff too. But at least from your study, we're going to be able to get at it from a fish and a macroinvertebrate perspective and see what it says about those two sites. So, we got this whole tangled web leading to biological integrity. Maybe we can just imagine, how do all of these different things affect the macroinvertebrates? So this mayfly, how are all these different things going to affect them? When we look at our samples today and we see what was there on the bottom of that stream in the server sampler, like, which of these things are going to determine who's here, right? Who's here and who's not here. All right, so you don't need to know these. I just kind of wanted to show you that, like, when we're talking about macroinvertebrates, it's a lot deeper taxonomy. So mayflies have been around since the Devonian period. So we're talking on the order of 400 million years. Like, these are really ancient groups, right? They've been around a long, long time. So we're talking about different phyla, different subclasses. So the mollusks, right? What are the mollusks? What might we see today that are mollusks? Snails, possibly some clams. There are these things called corbicula. They're little invasive clams from Asia. Everything invades from Asia. Why? I don't know. We might see some of those mollusks. Annelida, subclass Oligochaeta. What are the oligochaetes? The worms, yeah. The segmented worm, the few bristled worms, Oligochaeta. Yeah, so earthworms. There are aquatic earthworms that we'll probably see. We usually see some of them. They'll be in there. Platyhelminthes. It's like zoology review, isn't it? It's awesome. What are the platyhelminths? They're the flatworms, right? So we might see some leeches, for instance, right? Hyridinia. We might see some leeches from the flatworms. But what we're going to see the most of are, of course, the arthropods, right? So arthro referring to jointed and poda legs, so like the joint-legged animals. And so within those, crustaceans and the insects are what we're going to see, and the vast majority of what we're going to see are the insects. But can we think of some crustaceans that we might see? So water penny is actually a beetle, believe it or not. It's a beetle larva. Crazy, right? Yeah. So so they're actually insects, but there are a couple of crustaceans we might see. Might see some cryodos, might see some little ones, and we didn't record the ones that we saw with our electrofishing, but they're out there. Might see some young ones. There are also what are called amphipods. I'll show them to you if we see them. They look like roly-polies. You guys know what roly-polies are? They look like flattened roly-polies, like laterally compressed roly-polies, and they swim around. And they're shredders. So we might, although they won't be swimming around now. They swam their last swims a few weeks ago. But we might see some of those. Interestingly, crustaceans really abundant in the tropics. Shrimp play a huge role in stream ecosystems in the tropics, not so much up here. But the vast majority, of course, are going to be the insects. And this is why, right? So if we look at relative proportion of all described animal species, this piece of the pie is everything non-insect. This is everything insect. So, it just kind of makes stands to reason. In fact, globally, over about half of all the described species are beetles. You know, not so much in aquatic environments, although they're there, but terrestrial. If you find an animal and pick it randomly out of like all the animals, it's probably going to be a beetle. So yeah, so insects by far the most diverse. So I want to talk just a little bit about the basic body plan here so that we have some common language when we're in lab today. Moving from the top to bottom. So this top part here, that's kind of the easy one. This top section here. Not everyone all at once. Sorry, actually all of them. Yeah, head, thorax, abdomen. So you're going to see some things in your key today talking about the characteristics of the thorax, characteristics of the abdomen. And so these are just the three main body segments that make up the insect body plan. So when you draw this, be sure to get like these little like filaments like coming off this part of the arm. These ones coming this other way. The eyes on the side. No, I don't know. Yeah, so head, thorax, abdomen. And there's a couple other parts that we need to know about. So first, these are what are known as the terminal filaments, or they're also known as the cerci, C-E-R-C-I. This is something that your identification keys will talk about. So that's something you want to be familiar with. These structures on the dorsal surface of the thorax, what are these here? Anybody want to guess? Remembering that these are larvae of something, of course. Close. The gills are off to the side, on either side here of them, with the extra structure on the top. What do these kind of look like? Kind of look like wings, yeah. So these are actually the wing pads. So again, these insect larvae are, of course, just the immature form of these organisms. So they have to develop those wings as larvae, and in the pupal form, they'll complete their metamorphosis and then emerge. But the shape and the location of these wing pads are going to be important, particularly for mayflies, stoneflies, and then dragonflies and damselfly larvae. So wing pads up here. Brian, what are these here on the side? Those are the gills. That's correct. Yeah. So the gills are these sort of filamentous structures, and the location vary with order. But some keys will ask you about those gills. Now you'll notice that the gills in this particular invertebrate are, as we said, along the thorax. And you'll see why that's important in just a minute. I'll show you where there are other locations as well. So you want to be thinking about where, you know, where the wing pads, what do they look like, where the gill filaments, what do they look like as well. All right, so let's just take a look at the orders. So these are the 5 orders that are going to be most common. Ephemeroptera. What are the ephemeropterans? What's the common name for the ephemeropterans? I heard somebody whisper it silently, as if they were a mayfly. Yes. So ephemeroptera, those are the so I'm just defining these, but then we're going to go through the pictures and match them up. But so ephemeroptera are the mayflies. This is not a mayfly. Plecoptera are the stoneflies. Trichoptera are the caddisflies. And I would expect to know these, but Odonata are, of course, the dragonflies and damselflies. And then Diptera are what we call the true flies. So those include lots of stuff. So yeah, so these are going to be the most common insect orders that we'll see today. So looking at this one right here, anybody know what order this one belongs to? This is Odonata. What tells you that it might be Odonata? Say that again. No, you're right. I agree. And probably everyone looks at it and sees different things. But the actual defining characteristic, I think I shared this with someone the other day, is this labium, which is this kind of spoon-shaped feeding appendage that's like on the ventral side underneath the chin, and it extends this out to grab prey and bring it back. It looks something like this. It's pretty ferocious. These are like the tigers of the macroinvertebrate world. So this is a lateral view. This is a dorsal view from the top, and we can see there's some tiny little wing pads. But they have these big compound eyes on the side of their head, and they have, at the tip of the abdomen, they have just these sort of short appendages. Where are the gills in these guys? I don't see any gills. They're actually inside the abdomen. So middle schoolers love this when you talk about odonates and you say like they breathe through their butts, because it's true. They pump water into their abdomen through their butt, basically, at the tip of the abdomen. So they have these kind of rough appendages at the tip of the abdomen, and their gills are actually internal. So you don't see any gills on the outside of these at all. But the key is this labium that's underneath that chin. That's really what is going to tell you that this is an odonate larva. And so this is a dragonfly. This is in the suborder Anisoptera. There's two different suborders in these guys that look pretty different. So this is Anisoptera, the dragonflies. And then the other one is the damselfly, which is in the suborder Zygoptera. And so you can see the body shape looks pretty similar, no gills, anything. But they have these three appendages out here, which, if you look closely, actually look like feathers. And so the damselflies actually have their gills at the tip of their abdomen. So nothing along the abdomen, nothing on the thorax. But the damselflies have these feather-shaped gills at the tip of their abdomen. So Odonata, no gills on the abdomen or thorax. In the damselflies, they're on the external side with these feather-shaped gills. And with the anisopterans, the gills are internal, so they actually pump water into their abdomen to be able to breathe. And these are predators. These are scary. If you're a small invertebrate, these are scary. There's no other way to put it. Be afraid. Be very afraid. All right, this one here. What order are we thinking on this one? Yeah, stonefly, which is which order again? Plecoptera. Good. Yeah. So why? What makes you say it's a stonefly? So two tails. So here's the thing. There are also some mayflies that have two tails. So all stoneflies have two tails, but mayflies can have three, or in some cases two. So you got to be careful with that one. If you see three, then you know it's not a stonefly. If you see two, it's a good chance it's a stonefly, but it might not. It might be a mayfly in disguise. So really, the best thing, way to tell the difference is to look at the gills. And the gills are on the thorax, and that makes it a stonefly. So the body shape is pretty similar to the mayfly, but the location of the gills up here in the thorax. Those thoracic gills. What about this one? So it's got a similar body shape. It's got those wing pads on the dorsal surface. It's black and white. I'm sorry, it's kind of boring. But where are the gills here? Abdomen. So this is the mayfly, Ephemeroptera. It has abdominal gills. Now, some mayflies will have those gills going all the way down each abdominal segment. Some will have what we call opercular gills. They'll have like a flap covering them on the first couple abdominal segments. And they might be up close to the thorax, and you'll think they're on the thorax, but they're not. They look like, they're called opercular because they have covers like a fish gill does. However, remember the stonefly has gills right here, almost looks like in their armpits, like on the thorax. But the mayfly does not have gills on the thorax, gills on the abdomen. And if you see those 3 terminal filaments, then it's definitely a mayfly. C2 could go either way. All right, what about this dude? Good, this is a caddisfly. Awesome. What makes you say caddisfly? Yeah, so there was a stone house. And sometimes you find these in their little houses. So definitely the caddisflies, some of them are case makers. And they'll make their case out of all different things. Some make them out of silk themselves. Some make them out of sand. Some make them out of leaves. Some use twigs. So there's several families of caddisflies that are case makers. But often you get these without the case. Either they've come out of their case, or they're what we call free-living caddisflies. So if they're free-living and you just see this, what might tell you that that's a caddisfly? Any thoughts? So, yeah, it has a very long, soft-bodied abdomen, and so these humps on the side, yeah, those humps kind of help them stay in their house for the ones that are making the houses. So yeah, definitely that. And they've got this long, soft abdomen, and the main thing is at the tip of their abdomen they have like a hook. It's called an anal proleg and a claw, and they'll use that to stay anchored wherever they are on the substrate. So if you're looking at something and you're not quite sure, hard to say, look for that claw, that hook at the tip of the abdomen. That's the key for the caddisfly. Now, when you see enough of these, you see other things too. They have a longer, softer, curved body. It's not as armored as that of the stonefly, the mayfly. Those are trichopterans. Cool. And then we have the order Diptera, which is huge. I mean, it's huge and challenging to identify. So the dipterans are distinguished by what are called prolegs. So they don't have, as larvae, true segmented legs like all of our caddisflies, stoneflies, dragonflies, damselflies, mayflies do. But they've got these little nubs. So what you're probably going to get the dipterans confused with is like an oligocete worm. But what makes them different from an oligocete worm is a clear head and these little prolegs, these little appendages, which of course in the adults will become the actual legs. So these little things called prolegs. And they'll have little appendages toward the end as well. And this midge larva here uses these to breathe. This is a cranefly larva, which are starting to come out now because they break down the leaves. And we're going to talk in another class about the functional roles of invertebrates in ecosystems, and they do a ton of stuff. This is a cranefly larva. You ask, well, where's the head? I don't see a head on that cranefly larva. It is actually retracted into this sheath, and when they feed, that head will come out and feed. So sometimes the head is not easily apparent, but you'll see like a hole at the end where the head will be, and you can kind of see it through the color, through the skin a little bit. And then again, we have at the tip of the abdomen some little bumps and nubs, which are those prolegs. So like if it were an oligocete worm, or even something like a nematode worm, no head going on, no prolegs, no appendages at all. So with these dipterans, they look worm-like, but they have a head and they have some appendages attached. In the lab today, I'm going to give you all actual written dichotomous keys to go to order when you're identifying things, and then also to go to family within each order. So we're actually going to identify these invertebrates to family. But the first step, of course, is to go to order, so you'll know which key to bust out. All right. So there are a couple other things you might find. Might find some coleopterans. You might find some helgramites, those kinds of things. We'll see a couple of those, but these are probably the majority of what we'll see. Any questions on these families, the ID? I'm hoping we'll see some cool stuff this afternoon. Hopefully Bee Tree Creek. Bee Tree Creek, give us some cool stuff. because Sweeten Creek was, I don't know. We'll see. You find these little midge larvae a lot in lower quality habitat. So there may be some of these, and these can be tiny. So we might not have seen them when we were actually looking at the jar of the sample potentially. All right, awesome. So these insects are really funky because most of them have a larger larval than adult stage. And so just the life cycle for if y'all are not aware, involves this aquatic larval stage. And this is a mayfly. So the mayfly has a couple stages that not all orders have. But the larvae that we'll catch, of course, are the immature nymphs. And they will ultimately crawl out of the water. You'll see sometimes on stones near streams, they look like really big aquatic invertebrate larvae, but the back's kind of been like split open. And they look like hollow shells, like those cicadas that you'll see. It's because these things will crawl out of the water, then emerge, splay their wings out, let them dry off, and then once they've dried, they'll fly away. They'll fly away, they'll mate, and the female will come and drop those eggs back into the water. Those eggs will sink to the bottom, of course, they'll hatch, and the life cycle starts again. And this is a really neat evolutionary strategy, right? That if the water is safer for you than the land, then you can develop your larva there where it's safer. So there's this cycle. So just looking at how these insects have evolved to live in the water, it's kind of cool. So just looking at Ephemeroptera, which are the most ancient order, at least based on our current, most ancient order of aquatic insects based on our current knowledge. It's a couple different families. Heptageniidae, these are those grazing mayflies. Beatidae, those are mostly collectors. They go around looking for pockets of organic matter. And then Ephemeridae. How have these body plans evolved to allow them to live in lotic environments? What are the different strategies here? Any thoughts? So these ones right here are definitely flat, right? So they can stick right up next to a rock, and they're not going to get washed off easily. These guys are not so much flat. What do they have going on? By the way, you can really see those abdominal gills in this picture, right? Here they are, and here they are. Do you think this is going to crawl around the tops of rocks a lot in high flow velocity water? It's got more of a cylindrical body shape, kind of like a trout or a minnow, right? So this is a swimming mayfly. So the other thing you can do is you can have a streamlined body for being up in the water swimming around. So they've evolved two ways. And there's one more way you can evolve to deal with high flow velocity. Ephemeridae, anybody know what this guy does? It's got real stout legs right here. Almost looks like, you guys ever see like the camel crickets and stuff with the real thick, strong legs? What are they doing? Besides scaring the heck out of you when they come out of like a cave or your basement or something. They're burrowers, right? So the other thing you can do is get out of the water completely. And so these are burrowing mayflies. And so they use those real stout legs to burrow down into the sand and the substrate. And then, as I mentioned the other day, they actually create a water current and they filter feed out of there. So these macroinvertebrates have evolved different ways to deal with high flow and to be able to live in these rivers and streams. So Heptageniidae, these are grazers that are on the surface of rocks because that's where they feed. So they've got to be real streamlined and aerodynamic that they don't get blown off. These beaded mayflies, they're cruising around looking for pockets of food and organic matter. So they got to be streamlined like a fish to be able to swim. These ephemerids are filter feeders. And so what they do is they set up shop in a part of the substrate that's a fine part of the substrate like silt or sand, and they burrow in there. Place that probably has a little slower flow to begin with, and then they're able to hang out and just filter water through that tunnel, if you will, to feed that way. So a really interesting thing that people discovered about these aquatic invertebrates is that they drift. And this was initially found out when people were trying to figure out like how many terrestrial bugs that fall into the streams contribute to the food chain. Like how important is that? So I put out these nets to sample drift. But what they found were a lot of aquatic stuff drifting in these streams as well. So just to give you an example of how much of this occurs, a study in Oregon found 5,000 just caddisflies in 24 hours coming through one of these nets. 5,000. And the benthos density there was about 7,400 per square meter, right? So 1 meter squared. So almost the whole amount of caddisflies as would live in a square meter of the stream bottom was flowing downstream past this thing during the day. So what would you be interested to know about this? Why do you think that would be happening? Okay. Yeah, so maybe they're trying to disperse and it's just the most efficient way to move, Yeah, what else? Could be following the food, yeah, maybe trying to seek out greener pastures if you're a Heptagenia mayfly. Seek out greener rocks. The rocks are always greener on the downstream side of the fence. That's what they say. So just a couple thoughts. So they collected data on this, and it turns out that there's a lot more drift that happens at night. So these are data on Betus. And so, yeah, like right around midnight, a lot drifting, and then it drops back down again. This is what we call diel periodicity. Why might this pattern be going on? Any thoughts? Yeah, if you're avoiding sight-feeding predators, remember those the North American minnows, they've got big eyes, sight feeders. A lot of them have mouths at the end of their head, so they're going to be, feeding on things at the same level as they are. So a lot of them are drift feeders. So if you can come out at night, maybe you're safe that way. I did a study when we lived in Oklahoma for my PhD. It was kind of a pilot study, and I looked at this minnow called the Big Eye Shiner, and it was interesting. I looked at the gut fullness over a 24-hour period, and their stomach's actually most full at 3 in the morning, which is crazy. So there's going to be an interesting, I don't know, there's an interesting coevolution thing here. I don't know if maybe they're good enough sight feeders that some of them can feed at night. But the main hypothesis for why they do this is that there's a lot more minnows feeding during the day. And so this is a plot of drifting density compared to light. So this little rectangle are areas of very low light. And these are the numbers on the rocks, if that makes sense. So they looked at the numbers on the rocks versus in the drift. And so at night, not too many on top of the rocks, but a lot more drifting. And this is kind of a cool experiment. So people manipulated the light-dark cycle in experimental streams, and the invertebrates responded. So when they shortened the day and changed the timing of the day, they still came out and drifted when it was dark. So this is, you can't see that. So this is a behavioral, not an evolved response. So these invertebrates are entering the drift in response to dimming lights. So that's kind of what you would expect if they were doing it to make sure they did it at a time period that was dark. So interesting. So why? What are we getting at here? So this study looked at density in the substrate of invertebrates and drift of invertebrates. And then they looked at stream channels that had low amounts of food, which is this one up here, versus high amounts of food. And it was paraphyton because these were grazing invertebrates. So density of invertebrates versus drift of invertebrates on low level food patches and high level food patches. What do you think they concluded from these results about invertebrate drift. Yeah, trying to avoid competition, right? Because as the number of invertebrates goes up, the propensity to enter the drift goes up. And when you've got less food, you're more likely to enter the drift. So the main hypothesis about this is that drift is an active behavior in response to low light to try and find more productive patches. So a question, maybe crystallizing in your mind right now. If these invertebrates are always drifting downstream, why don't upstream areas eventually have no invertebrates left? That is the predominant hypothesis, yes, that the adults fly upstream, lay the eggs, and then as density rises, they keep drifting downstream. So, anyway, interesting thing about. So there's kind of three different types of macroinvertebrate drift. There's what's called passive. So there's some invertebrates like midge larvae or one, they kind of drift at the same amount all hours of the day. So that's probably a passive, just kind of entering the drift. You get too close to the top of the rock, and whoa, where'd Bob go? Oh, he's downstream. Okay, whoops. But then there's also catastrophic drift. So, for instance, after organic chemical spills or fertilizer spills, they found really high levels of invertebrate drift, where they're either injured or they're just trying to get out of there. Essentially, like they have to leave. Yeah. So rain event, that'd be an example of catastrophic as well. And it's interesting because like a flood would have to be really extreme for it to be this, because this is something that would really affect the population. But if it was a big enough flood, it could. Yeah. Yeah. We're finding in some streams that the water can get up a lot higher than you might think. And then this final piece is behavioral. So the idea is like to avoid predators and try to find greener patches. You enter the drift. Yes. Yeah, they could have. They could have. So that's one of those things where, generally like daytime drift is pretty low. So compared to whatever's on the bottom of the stream, like that input should be pretty low. And we would just hope that like it was random sort of across our different samples, So like, yes, I'm sure we did have some that drifted in from upstream, but as long as like this silver sampler didn't have a ton that drifted in and this had none, then hopefully that'll kind of even out across the replicate. So yeah, and it's tricky because you get more invertebrates where it flows higher. So you might say, oh, maybe more drifted in. But also those high flow habitats are better habitats to begin with, So even if it's flowing really high, you probably get more just because the habitat's better. And that's where, oh my gosh. So for my dissertation research, for my benthic samples, I use this thing called a vacuum benthos sampler. It's a pain in the butt. It's this big like stove type thing. It's got these two handles. You kind of like work it down to the substrate. Then you have this pump that was a pain to build, but it like sucks water in through this tube and then through a net and then back out, and it like circulates. So you like isolate a particular part of the stream and then just disturb the substrate, and you don't have to worry at all about drift coming in. The other thing is I had to do that because I was sampling some places that were high flow and some places that were pools. And of course, a service sampler is not going to effectively sample a pool because you disturb the substrate and the bugs just go everywhere, right? They don't go into your net. So there are ways to sample where you completely eliminate that. But since most of our sites had enough flow to get the invertebrates in there, and we were doing it during the middle of the day, you'll find that Cerber samplers are a pretty standardized method. So you just kind of assume that whatever drift is going on is going to be sort of spread across all your samples. But that's a good question. It's a good question. All right. So you all have an idea of kind of how macroinvertebrates vary over spatial scales of like tens of meters, right? Because you sampled riffles at Sweeten Creek, you sampled pools. You're kind of familiar with some of those differences. But let's take a little broader view, right? Some broad scale geographic patterns. So these authors looked at richness for 3 of these orders, and they looked at 495 published studies. So they looked at a whole bunch of papers that document a number of genera. Why do you think they didn't use species for their study? Any thoughts? Could be too many, a lot more. As an aside, I will note that we identified fish species, 2 species, but invertebrates, we're going to identify a family. Man, it's just a pain in the butt to identify invertebrates to species, as folks who've taken Dave Penrose's course know. So I think when they looked at genera, you're a little more certain about the data. You can tell it's a genus, but going to species, like, man, it's a pain in the butt. Okay. How are you guys doing? Do you guys need like a minute or two to stretch? You look like kind of like low energy. Okay. Take like a 2-minute stretch break. Like input. Or do you want, okay, why don't we go ahead and make it, yeah, let's go and make it like a 5-minute break. And if you need to go and take a short walk to the restroom, you can go ahead. What's that? Meet Tuesday, the new Monday. Oh yeah. Reasonable, right? Like, what does that mean? Like p-value of 0.05? Like, what all? Let me talk to you about sampling efficiency first, and then we'll get to that. Yeah. Let me talk to you about the latitude diversity gradient. So this is kind of cool. I don't know. This is like one of those mysteries, right? That like, I don't know, Darwin and other dudes and dudettes were curious about. Like, why are there more species in the tropics? So this is like for terrestrial vertebrates. It looks like the Amazon is, or part of the Amazon is on fire, which it actually sort of is or was. So there's this latitudinal diversity gradient, which y'all may be familiar with. And it sort of holds true for fishes as well. This is, these are darker colors. Darker colors equal more fish species here on this graph. So the latitudinal diversity gradient kind of holds true for freshwater fishes as well. So these authors asked the question, like, what is it that determines diversity of aquatic insect species around the globe? So, like, what are some of the hypotheses that we have for explaining this pattern in latitudinal diversity gradients? Anybody know? Warmer? Okay, yeah. So how does warmer equal more species? More growing season? Direct sunlight? Okay. Yeah, so there's this idea that like higher productivity can lead to more species, kind of for a couple of reasons. One of which is that, well, that's a good hypothesis. I'll go over those too. Other hypotheses? Say that again. Refugia, yeah, from like glaciers. And were you thinking other disturbance as well, or glaciers in particular? Good, yes, all true things. So just a couple. So one of these is productivity. How might higher productivity lead to lower extinction? To respeciate. So could be, so speciation usually requires like a separation event. But one thing, what's true about our population sizes when productivity is higher? thinking of the good old pyramid of numbers, right? When we start with higher productivity at the base of the food chain. Goes up, yeah. We can support larger populations. So in any place, any of these places on the planet, the balance, but there's a balance between extinction and speciation events, right? So places where extinction events are lower should have more species overall. Why are you at risk of extinction if you're in a smaller population? Less individuals reproduce, yeah, and how might extinction kind of happen? So you can increase your population faster if you have more individuals reproducing. And from the perspective of extinction, how might you be less vulnerable to extinction if you have a larger population size? Yeah, and that might be related to what you mentioned as well, Michael. Yeah, so the gene pools are more genetic diversity, so the ability to evolve in response to differences. And another thing that can be important is random events. So like disease, predation, disturbance, all those things we have consistently been shown like wipe out smaller populations faster than large populations. So areas that have higher rates of productivity lead to higher population sizes, lead to more resistance to extinction. The area hypothesis. So if you imagine islands and they go from smaller to larger, starting with like a tiny little island like the funny Far Side cartoon where there's like two dudes on a sand island with a palm tree and then one of them says something funny, which I don't know how funny it would be if you're really on a desert island by yourself. But you imagine starting with that and then islands getting bigger and bigger and bigger. And as those areas get larger and larger, they include more kinds of habitats, more niches, and of course larger population sizes, so lower extinction. So in the tropics, we have higher productivity, because just to review, what are the variables that determine productivity on land? One was mentioned already. Sunlight and water, right? Sunlight and precipitation. They drive productivity on land. So these areas having high productivity, more species. Greater area, what's interesting is that if you imagine latitudinally the weather getting cooler towards the equators, right? So there's an area that has similar temperature and things as where we are, in the same amount of degrees south latitude. Does that make sense? So go from warmer to cooler, warmer to cooler. Okay, so similar temperatures here as here. But those two habitats aren't connected. They're very far apart. Well, around the equator, all of these habitats are connected one to another, right? So the habitats, as you move north, as you move south from the equator, these areas are connected. So there's some thought that in the tropics, because those habitats are connected, whereas habitats at higher and lower elevations are similar but not connected, then you have higher species diversity there as well. And then the disturbance fact that Torn mentioned, when we look at higher elevations, there's this history of disturbance, which is problematic for lots and lots of species. And so because of that, then at more extreme latitudes, we've had higher historical extinction rates. So, these are kind of the main three hypotheses that the ecologists use to explain this latitudinal diversity gradient. It exists for lots of species, not all of them. There are a couple exceptions. But, in fact, freshwater mussels, unionid mussels, and salamanders actually almost higher than anywhere else in the southern Appalachians. But for a lot of species, we have this gradient. So these authors asked, okay, where is insect diversity highest and why is it? So before we do that, it's always tricky to see through the Zoom box. But it's always tricky when people do these sorts of meta-analyses. In other words, they're not collecting their own data. Their research trip consists of like going to their computer and like the Web of Science search engine. Doesn't that sound like fun? Yeah. So these sorts of analyses kind of have they have issues. So these authors themselves mention some of these. Completeness of data, right? In spite of the fact that we compiled a much larger data set than previously examined by others, and we used the statistical models that best fit the distributional properties, the most significant limitation in the data we compiled was the paucity of information for streams either near the equator or the poles and for certain biomes. So you run into this problem of certain areas being studied more than others. So you just kind of have to be upfront about this. There's been a lot more research done on the Southern Appalachians than tropical streams. Not because people don't want to go to the tropics and study those streams, it's just harder to do. And you can take a class out and, we've got lots of universities here in the Northern Hemisphere. So keeping that in mind. So they looked at these different biogeographical realms, the Nearctic realm, the Palearctic, Neotropical, Africotropical, Indo-Malayan, Australian. And then these dark spots are locations of the studies. And so you can see, right, just not that much work that's been done in South America, sadly. Not as much in Africa, relatively speaking, a lot going on in North America and Western Europe, perhaps not super surprisingly. Yeah. Yeah. So interesting question, and I'd have to research that more to get back to you. But I can say that we have ways of statistically getting at that, of like estimating diversity, and it has to do with the principle that well, this may be another conversation, but essentially like when you first go out and sample a site, let's say you get 10 species, right? The next time you go out and sample that site, let's say you get, 8 species, but one of them's new, you didn't get before. The next time you go out, you get 11 species and 3 new ones. So you can plot the rate that you accumulate new species, if that makes sense. And so you can plot what are called species accumulation curves. So the short answer to your question is, I don't know, but there are ways to get at it that we should research. I've used some of them sometimes. Okay. So they looked at these patterns for macroinvertebrates. And so here we go from the equator to north latitude and to south latitude. And this is a plot of the number of species. Ephemeropterans, Plecopterans, Trichopterans, and then EPT in general. So what do you see here? If you, get your data back and you look at this on the bottom panel, what are you concluding about where diversity is highest? Data's never as neat and easy as you'd like it to be. You got to squint at it and then run screaming into the night a few times and then come back and then in the middle of the night, usually you wake up, you're like, oh, that's what the pattern is. Oh my gosh, I get it. But if you just were to look at this, where would you say diversity is highest? Yeah, north latitude, right? You are reading it right. Yep. So at about 40 degrees north latitude. And there's this interesting little trough here between the equator and 40 degrees north latitude. So there's our equator. So if we look at the areas of highest diversity, it's this section right here in the northern hemisphere. Also the area that's been studied the most. But remember, each of these dots is a study. So even these studies done in the tropics near the equator, weren't, some of them didn't get many species at all, right? So we do have some data from there, so we don't have a ton. So based on the data that we have, yeah, there's you are living in an area of very high aquatic insect diversity. Isn't that cool? Nice. All right. But what kind of puzzled them was this little trough right here. So they looked at a couple things. They looked at terrestrial productivity. They looked at latitude, and there's just a lot of noise here. So this is the equator. These are those tropical sites in the green. But I don't know, man, if you were to plot all these data and draw a best fit line, so we have latitude from the equator to north latitude, latitude from the equator to south latitude, and this is a measure of mean annual productivity. But I would like to see a best fit line, and it'd probably go something like this, or like maybe this. But there's not a huge, you know, there's areas of high productivity everywhere. Ton of variation. So latitude doesn't correlate super well with productivity on these data. Basically, all you have is that in the tropics, you don't have too many areas of low productivity, right? But you've got areas in northern and southern hemispheres that are just as productive as the tropics in certain places. Then they looked at this. They looked at flow variability in rivers and streams in this area. They looked at the coefficient of variation. It's how much variation you have divided by the average, and then multiplied by 100. Look at these peaks, right? Rivers here at this in this area, 20 degrees latitude, lots of variable flow. That's something that, if you're a terrestrial species, you don't really care about that much, generally. But if you're an aquatic species, you might. So then they came back and they looked at these areas of latitude back on their figure, and those areas of lowest species diversity is this area right in here. And there's a lot of variation here in the flow. So yeah, you've got the monsoon impacts over here. I'm not sure why. Does anybody know why Mexico and Central America has really variable rainfall patterns? I don't know. They don't have like a monsoon, do they? Or some kind of, I don't know. But anyway, this area, really highly variable flow. So plenty of productivity there, but really variable rainfall. So that appears to be driving diversity here. There's a lot of data. That's kind of scary the way it came up like that. I shouldn't have done that. I'm scared now. So then they looked at like annual productivity versus genera diversity, and there's kind of a positive relationship here. I just look at these two. That's all you need to look at. So there's a positive relationship between net productivity and number of species, and a negative relationship between variation in precipitation and number of species. So if you want to find the place with the most species or genera of aquatic macroinvertebrates, find a space that has really high terrestrial productivity and really low variation in flow. So those kind of explain the patterns in insect diversity that we see. So I wanted to mention just a little bit about conservation of aquatic invertebrates and why it's so challenging. So this was written by David Strayer, who you might remember. He wrote that article on tinfoil covered rocks being used to study freshwater mussels. And he pointed out a couple things. The first thing he noticed is that when we look at the number of described species, these are all invertebrate groups. These are fish. There's a lot of invertebrate species that have been described relative to fish. But when we look at the number of papers on them since 1990, this is the area that he looked at, there are a lot more papers. And so this is a log scale, right? So this is 10 times as much as this. Sorry, yes, 10 times as much as this. So the take-home message is that there are total many more species, particularly of insects, than fish, but they've been understudied, right? We have more papers on fish than other things, which, you know, I think, whatever, like, that's cool. But, like, no, like, we need to study invertebrates more. Now, even if you look at species that are legally listed, we study vertebrates a lot more than invertebrates. So there's been this sort of study gap on invertebrates. And then just a couple of factoids for you. The extinction rates of freshwater animals in North America. This is freshwater organisms as opposed to terrestrial. Species loss in aquatic habitats, freshwater habitats, is 5 times that for terrestrial habitats. So we're losing aquatic species faster than terrestrial ones. Just as an example, mussel species in the Mobile River Basin, at one point the most diverse in the world, about a third of those species are gone due to flow regulation and habitat alteration. And then we have this whole problem that in latitudes that are not temperate, we don't really know. Like, we don't know what's going on with aquatic species, right? So this is kind of the problem with aquatic species. We're losing them faster. We've lost a lot. We don't really even know what's going on in these non-temperate latitudes. And so this is kind of part of the reason that we wanted to do our study in Nepal. And so I wanted to share just a little bit about that with you. So we were studying this river basin in Nepal, as I mentioned, the Kali Gandaki, Narayani, and there's a conservation area here, conservation area down here, and then these are all sites that we studied, and they varied in elevation quite a bit. I thought I'd show. Remember, I talked about that place where they created this wall to keep the river from flooding the village. This is an aerial view. This is that wall right here. This is the river coming in here, and you can see where it used to go in there, right? You can see this old channel. It used to just flow freely through there, but now there's this constant wall that's being built to keep it from getting back into this village. And so this is, you know, a reservoir impounded here and then downstream here. This is what the river looks like from upstream. This is, of course, a dam that's built across. This is what it looks like from the downstream side. And then this is a view of that wall. So, like, I don't know, these are about 3 to 4 feet tall. They're just all these giant cement blocks that are built, and they're kind of in this process of constantly falling in and being built anew, as I mentioned the other day. And so we looked at all of our sites, and in the 1980s, some of these downstream sites, so the area I just showed you is right on the bottom here. Some of these downstream sites had between 20 and 30 species in the 1980s. In the 2010s, this is species change. And so red means that they lost 20 species, and orange means that they lost between 13 and 19 species. And so these areas way downstream in this section have lost a bunch of species. So I just bring this up to make the point that, like, in non-temperate areas, like, it's the same problem. We're still losing lots of species. And so just a couple things, and I'll end on this. So Strayer brings up 3 points that make conservation of freshwater habitats challenging. But let me ask you, what do you think it might be about freshwater habitats that makes them more challenging to conserve than terrestrial? Yeah, absolutely. You've touched on a couple things there. Yeah, yeah, good. Any other thoughts? So I mentioned a couple things. First of all, they're scarce. If we look at the landscape, there's a lot more terrestrial habitat here than aquatic. There just is. If you were to add up the square acres, a lot more terrestrial than aquatic. So there's a lot more places for terrestrial species to go to. But all these freshwater habitats are essentially islands, right? They're separated from each other. And so, like we mentioned, if you want to go from the Swannanoa to the Catawba River, geographically it's close, but fish can't hitchhike up I-40. They got to go all the way down that network to the ocean and up, and they can't do that. So all freshwater habitats are islands in a sea of either terrestrial habitats or saltwater habitats. Some of y'all may have taken Landon Ward's course on conservation biology, and I think he talked about this idea that area is a key determinant of how many species you can protect in protected areas. And whether it's good to have several, a single large or several small habitats. Well, in freshwater, all we got are several small isolated habitats. And then what Brian mentioned is freshwater habitats are the downstream end of all the terrestrial habitats, the downhill end. So everything that happens that's bad in the terrestrial realm gets washed into the stream. And then because of the fact that all these freshwater habitats are essentially islands, those species don't have anywhere to go. The only way they can move is somewhere else in that stream network. Right? So if we have land that gets polluted somewhere, those birds are going to be able to fly and find other habitat. They're going to be able to move around. But with aquatic organisms, they don't have that option. And so these three things are factors that really make freshwater habitats imperiled. So yeah, as I was putting this lecture together, I kind of started on aquatic macroinvertebrates and ended up talking about conservation of freshwaters in general. But there were a few things that I realized I don't think we'd really talked about yet. So anyway, so this is why it's so important to conserve them. And this paper that documented these extremely high levels of extinction in aquatic habitats was published in 2006. There was just another review that came out this past year, and not surprisingly, the same problems are still going on. So we haven't been able to fix it yet. So some reasons to be concerned about freshwater habitats and be thinking about protection. I guess the last thing I'll say is that this is a national park right here, upstream from these dams. And the areas that we found the lowest reduction in species number were largely in that national park. So area that's protected, we really have to keep it protected because you can't undevelop the watershed once it's developed. Okay, awesome. So hopefully y'all saw the numbers. I don't know, MC, you were interested in maybe switching with someone? Okay, cool. So yeah, so we'll see half of y'all at 120, and then whatever the time was I put for the second group, we'll see the other half, all right? Cool. See y'all this afternoon. Have a fabulous next few hours.